feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lun School of Medicine Healthy Longevity webinar. It's a great pleasure that you're joining us at edition number 101. I hope that you enjoyed watching our 100th show, but if you missed it, it's all on our YouTube channel, The 100 Show, and all the other editions. Professor Passos is our guest, talking about one of the hugely studied hallmarks of aging, and that's cellular senescence. And he will talk about the relation with mitochondria. So it will be a very interesting presentation. Please use the Q&A function to send questions, remarks, comments, your thoughts, whatever you would like to put in the chat. All are welcome, and we are watching it. Before tuning to Professor Passos, Dr. Liuan Guan, our research fellow at the NUHS Center for Health Longevity, will shortly present on a study talking about blood biochemistry-based biological age in relation to frailty in a quite unique cohort of geriatric rehabilitation in patients, and that's a resort cohort. So, Liuan, go for it.
Thanks for your kind introduction. As mentioned, my name is Li Huan. Today, I'm going to share one of my projects entitled Higher Blood Biochemistry-Based Biological Age is Associated with Frailty in Geriatric Rehabilitation in Patients, the resort cohort. Generally, frailty is considered as a state of increased vulnerability to poor resolution of homeostasis following a stress. The incidence rate of frailty is high in older adults and has been associated with many adverse health outcomes. It's well known that individuals age at the same rate chronologically, but not biologically. Biological age could be measured by aging clocks based on multi-omics technologies. In 2016, in silico medicine company developed an aging clock by machine learning based on routine blood tests. Whether biological age measured by the aging clock is associated with frailty in geriatric rehabilitation in patients is unknown. We used the resort cohort. After acute care in the hospital, older and frail patients who had multimorbidity were admitted to geriatric rehabilitation wards for recovery. At admission and discharge, comprehensive geriatric assessment, CGA, was performed to collect data from different domains. Then we did 3 months and a long-term follow-up. Blood tests of 30 frequently measured parameters undertaken at admission were used to measure biological age. Age, sex, and the pathology data were input the aging clock model on Clock platform. It outputs biological age with the unit year. The determinant is delta of age calculated by biological age minus chronological age. The frailty phenotype was assessed using the Clinical Frailty Scale, CFS. The severity of frailty at admission was treated as an ordinal variable from mild to severe frailty. The change of frailty from admission to discharge was grouped into stable, improved, and deteriorated frailty. The effect modification of comorbid condition was tested. The median age was 83 years old. 57% were female, The comorbid condition is assessed using Cumulative Illness Rating Scale, CIRS, which quantifies the burden of illness across 14 physiological systems. A higher score indicates a greater comorbid condition. The median of CIRS score was 12, indicating 50% of patients had illness in at least 3 physiological systems. The median of CFS score was 6, which means moderately frail. The median biological age was also 83 years and strongly correlated with chronological age. The median delta age was negative 0.61 years. In total patients, every 1 year higher in delta of age was associated with more severe frailty at admission. In patients with CIRS less than 12, the association maintained significant, but not in patients with higher CIRS scores. Compared to stable frailty, delta H was not associated with either improved or deteriorated frailty, regardless of comorbid conditions. This is delta H in different frailty status, stratified by CIRS score. We can see delta H increase with the severity of frailty in patients with lower CIRS scores, but not that linear in those who had higher CIRS scores. Within each category of frailty, patients with severe comorbid condition had older biological age. Using the decomposed CIRS, we demonstrated that diseases affecting cardiac, hematological, respiratory, renal, and endocrine systems are more representative of the aging-related blood profile changes. More severe conditions, namely frailty manifestations, in these systems contribute to higher biological age. I would like to thank my supervisors, Prof. Andrea Meyer and Dr. Camilla Tuttle, and collaborators from In Silico Medicine, Dr. Alex and Fedel, for their support. And thanks to Royal Melbourne Hospital for data collection. That's all. Thank you for listening.
Thank you so much, Liwan. I think it's wonderful to see that in geriatric care now also biological clocks are being at least tested before being applied. But let's go to another hallmark of aging, and that's senescence. Professor Joao Passos is with us, and he directs the Cell and Molecular Aging Laboratory at Mayo Clinic. And he holds the position as Associate Director of the Robert and Arlene Kogok Center on Aging. And he is the Director of the Discovery Science Team over there. He really focuses on cellular senescence. That's the reason why we know each other. He does beautiful work on the mechanisms of aging with relation to senescence, in particular with regards to telomeres and mitochondria. He first worked at Newcastle University at the Institute for Aging in the UK, and then since 2018, he moved to the US to join the Mayo Clinic. His laboratory has been continuously funded very successfully by several UK grants, European grants, and now also US funding bodies. And he has published over 100 beautiful articles, which I think really lay the foundation of the senescence work. So it's an honor having you today here, Joao. It's really nice to see you again, but especially hearing your latest insights into this topic. So the floor is yours.
Thanks a lot, Andrea. I'm delighted to be here. So thank you for this very kind of invitation. And yeah, I really hope that today I'll be able to show you something new about cellular senescence and its relation with mitochondria. So I'll start with a kind of, with a kind of boring slides for whatever reason. I can't move my slides. Okay, here we are. So basically an explanation of what senescence is. So cellular senescence is a, complex phenotype, but it's actually essentially a stress response. So when you have cells that are exposed to different types of stresses. These can be oxidative stress, DNA damaging agents, inflammation, telomere dysfunction. These stresses mostly engage two pathways, the P16 and the P21 pathway. P16 and P21 are cyclin-dependent kinase inhibitors. Their role is to stop a cell in a cell cycle arrest. But actually, senescence is much more complicated than a cell cycle arrest. So the cells experience a variety of changes in terms of their morphology, in terms of their gene expression, in terms of their chromatin organization, in terms of their mitochondria. But there are two very important factors that happen in senescence that I want to allude today. One is the fact that senescent cells seem to upregulate mechanisms that render them resistant to apoptosis. So these cells are actually pretty resistant to apoptosis. The other thing is that senescent cells secrete a number of pro-inflammatory cytokines and chemokines that are collectively known as the SASP, the senescence-associated secretory phenotype. And it is thought that this SASP is a way for senescent cells to communicate with the immune system, potentially recruit immune system to their vicinity, and that way orchestrate their own clearance. So senescence seems to play good roles and bad roles throughout our lives. It is thought that senescent cells evolved as a way to stop cancers from developing. This is, tumor suppression. The idea is that, if you have damage in a cell, you don't want that cell to propagate. But more recently, senescence has been implicated in other processes, such as tissue repair, but also embryonic development. And what is common about senescence happening in these instances is that, these senescent cells arise in our tissues, particularly when we're young, and they're very fast at signaling to the immune system so that the immune system comes and removes these cells quite rapidly. So they don't stick around for a long time. However, as we age, we start realizing that senescent cells start accumulating. And in fact, they've been associated with a number of age-related diseases, including, diseases of the lung, like chronic obstructive pulmonary disease or IPF, pulmonary fibrosis, metabolic diseases such as NAFLD, type 2 diabetes, but also neurodegenerative diseases such as Alzheimer's and Parkinson's disease. The question is whether or not these cells are present in these conditions simply because, as a consequence of other aging processes, or if they are actually contributing to aging by itself. And the big revolution in the field was really the studies that were published by the groups at Mayo Clinic, Jan van Dersen and Jim Kirkland, that use mouse models where it's possible to eliminate these senescent cells. And what they found is that if we remove senescent cells from an aged mouse, we can basically improve a number of conditions during aging. And in fact, we can extend their median lifespan. This led to the idea that senescent cells could be causal drivers of the aging process. And since these discoveries, many groups have shown that if we eliminate senescent cells, either through genetic means in mice or through the application of drugs that can kill senescent cells, we can actually improve a number of age-related conditions, including cardiac aging, diabetes, fatty liver disease, and also neurodegenerative diseases. And this obviously led to a great, sort of revolution in the field, because we are now thinking that, we might have a way by targeting one single biological process to prevent many age-related diseases simultaneously rather than targeting one at a time. So how do these senescent cells cause tissue dysfunction? So this is a kind of, general wisdom where, senescent cells actually accumulate in very low numbers. And more recent data where we can actually look at single cell data sets, we realize that it is likely that the percentage of senescent cells that we find during aging is actually very, very low. But the reality is that these cells, through the SASP, are able to have widespread effects. First thing is that senescent cells can actually transmit senescence via the SASP to surrounding cells, thereby spreading senescence in tissues. Via the SASP, senescent cells can also impact on stem cell niches, thereby impairing regeneration of tissues. Senescent cells also produce proteases that can, degenerate the spaces in between cells, extracellular matrix, leading to tissue cellular disorganization. By recruiting immune cells, they can enhance, exacerbate inflammation. They also produce pro-fibrotic factors leading to fibrosis. And it has been shown that senescent cells can also enhance the proliferation of cancer cells, thereby, almost paradoxically, enhancing cancer. So for these reasons, the field has kind of opted for two different strategies in which we should, target senescent cells. One is based on the idea that senescent cells upregulate these mechanisms that render them resistant to apoptosis. So the idea is that if we target these mechanisms, we can kill off these cells from tissues, and then you'd end up with beneficial effects by removing these cells. And this strategy is usually called senolytic. The other strategy is really to target the SASP. And this is important. So because if we can stop the SASP, but at the same time preserve the cell cycle arrest, and I should point out it is important that we preserve the cell cycle arrest because we don't want these damaged cells to start proliferating uncontrollably. So the idea is that then the cells would remain, but we would stop the SASP that would cause damage in surrounding tissues. And what I would like to tell you today is a story that from my lab that kind of indicates that in order for us to be able to develop these therapies and understand these processes, we need really to understand mitochondria. So the mitochondria are central in these two facets of cellular senescence. So, you all know that mitochondrial dysfunction is a hallmark of aging. But mitochondrial dysfunction is also something that happens in senescent cells. And my lab and others have spent a lot of time characterizing mitochondria in senescent cells. And what has been shown is that senescent cells experience many changes in mitochondria. So one of them is that obviously mitochondria experience changes in their ability to fuse and to separate fusion and fission. So there's changes in mitochondrial dynamics, there's changes in mitochondrial mass. And if we look at the electron transport chain, where the energy necessary for the survival of cells is produced, we see that there are changes in the electron transport chain of mitochondria. So there's increased proton leak, there's decreased mitochondrial membrane potential, which leads to low levels of ATP, and ADP leads to low NAD plus NADH ratios. There's also evidence that mitochondria in senescent cells are not really, when they are damaged, the sort of mechanisms that regulate cellular homeostasis, like mitophagy, are impaired. So mitophagy is a way in which cells can basically remove damaged mitochondria. But again, while we describe all these changes in senescence, it was not clear if these changes are merely a consequence of the senescence program, or if in fact mitochondria play a causal role in senescence. So in order to answer this question, my lab decided to do a kind of crazy experiment at the time. So this was already in 2016. And we wanted to design a proof of concept experiment to prove if mitochondria were really necessary for senescence. And the way we could do that is taking some cells and overexpressing a protein called parkin. And parkin is a ubiquitin ligase that when mitochondria are damaged, parkin binds to the mitochondria and tags them and targets them for degradation by a mitophagy. And what we found is that it is possible to take a cell expressing parkin, and under certain conditions, if we treat it with a poison like CCCP, which uncouples the mitochondria, we can trick the cell into believing that all the mitochondria are dysfunctional. They are all targeted for mitophagy, and you end up with a cell that has virtually no mitochondria. And here you can see this is a senescent fibroblast, and this is a 3D reconstruction, 3D electron microscopy reconstruction of the cell. In red, you have the mitochondrial network. In blue, you have the nucleus. And these are cells 16 days after we had removed the mitochondria, where we cannot identify any organelles present in these cells. And if we look at expression of any mitochondrial proteins, if we run a Western blot, we see this empty band. So there are no mitochondrial proteins produced in these cells. And I should point out that these cells, even though they do not have mitochondria, they're still able to survive for long periods of time simply with ATP that is generated via glycolysis. So having established this model, we then decided to ask the question, what happens if we take a senescent cell and then we remove the mitochondria? So we did that, and then we looked at gene expression by RNA sequencing. What we found is that genes that were associated with proliferation were still down. So these cells were not able to proliferate again. But if we look at all the components from the SASP, the inflammatory factors, they went up in senescence. And then when we removed the mitochondria, they went back to the control levels, suggesting that mitochondria are really essential for the generation of the SASP. So this kind of proof of concept experiment led my lab to really be interested in understanding what it is about mitochondria that changes in senescence. So what are the mechanisms by which mitochondria contribute to the SASP? And also we started a drug discovery program trying to identify new drugs that target mitochondria with the idea of suppressing the SASP, and we call these drugs mitolytics. So what I'll show you now is one of the mechanisms that we think leads to this by which mitochondria contribute to the SASP. And this is a story that just came out yesterday. So it's a perfect timing. So it just came out yesterday in Nature, and that I'll be able to kind of break down to you. So we made the observation, and this was already, several years back, that if we look at mitochondria in young proliferating cells and senescent cells, and this is a staining for outer mitochondrial membrane protein, TOM20, and for DNA, what we found is that in a young proliferating cell, majority of the DNA will be present within the mitochondrial matrix. However, if we look at senescent cells, we found that there was a lot of DNA that was located in the cytosol. So if you look at this 3D rendering of a senescent cell, you can see the mitochondria form these tubular structures where the DNA is inside, but we can find areas where the DNA is sort of coming out or is already present in the cytosol of the cell. And we also found that this DNA, and I'm not showing here, is also associated with another protein called TFAM, which is involved in the packaging of the mitochondrial DNA. We also fractionated the cytosol of senescent cells, and we did this in many types of different cell types, but also different ways in which we can induce senescence, either by replication, replicative senescence, or by expression of oncogenes, or by exposure to DNA damaging agents. If we fractionate the cytosol of a senescent cell and measure the amount of mitochondrial DNA, we found that it's always enriched, consistent with the idea that the mitochondrial DNA is sort of coming out to the cytosol of senescent cells, but not proliferative cells. So what is the implication of this? So obviously, mitochondria were once bacteria, okay? They have their own DNA, their own circular DNA, which has many similarities to the sort of bacterial DNA. And it is believed that when this DNA is actually coming out, the cells react the same way that they would do if they would be infected by a sort of foreign DNA. And they cause an inflammatory response. So we decided to try to investigate the role of mitochondrial DNA in senescence. So we again went back to our model where we basically have cells that have mitochondria. Then we remove the mitochondria from a senescent cell using the Sparkin CCCP system. And then we have a cell with no organelles, but then we put back inside the cell only the mitochondrial DNA. And then we looked at gene expression. And what we found was quite remarkable. So if we look at genes that are inflammatory genes that are controlled by NF-kappa B, what you see is that in senescent cells, they go up. If we remove the mitochondria, they go down. If we put back the mitochondrial DNA alone, they go back again. If we then look at genes, these are interferon response genes that have been associated with mitochondrial DNA stress. Again, we see the same pattern. Up with senescence, down when we clear the mitochondria, up again when we reintroduce the mitochondrial DNA. Consistent with the idea that this mitochondrial DNA alone could be a driver of the SASP. We also did other experiments where we used a viral protein that is able, a DNase, that is able to degrade only mitochondrial DNA. And we can do that by expressing this protein in an inducible way, and we get very fast degradation of the mitochondrial DNA. And if we do that and then we induce senescence, we can look at the expression of some of the inflammatory factors associated with senescence, like IL-6, IL-8, IL-1 alpha. We can see that they go up in senescence, but then they go down if we degrade only the mitochondrial DNA, consistent with this idea. So why is this, how is this mitochondrial DNA basically inducing the SASP? It is doing so by binding to a DNA sensor called cGAS. cGAS will then activate STING, and STING is involved in the expression of both interferon genes and NF-kappa B. So we have shown that in senescent cells, there is colocalization between TFAM DNA complexes and cGAS. We've also shown that if we knock out using CRISPR-Cas9 both cGAS and STING, we can suppress the SASP in senescent cells, which is consistent with what others have shown as well in the field. But then the question comes, so if mitochondrial DNA is playing such a great role, why is this mitochondrial DNA leaking to the cytosol? What is the mechanism? And here comes the interesting observation that we made. So it had been shown in the apoptosis field that something similar happens. So apoptosis is very much dependent on mitochondria. And what happens is that there are some pores that are formed in the mitochondrial outer membrane by these proteins called BACs and BAC. These macropores allow release of cytochrome C, and cytochrome C then binds to a path, forms this apoptosome, and leads to the activation of effector caspases that ultimately lead to cell death. But it had been described in 2018 that the formation of these BACs and BAC macropores also allow the release of mitochondrial DNA into the cytosol of apoptotic cells. So we thought, could it be possible that something like this is happening in senescence? But we thought it was unlikely at the time because, as I mentioned to you in the beginning, apoptosis and senescence are usually seen as two different things, two different cell fates. And in fact, senescence is something that is thought to be resistant to apoptosis. So it is unlikely that such a process would be occurring in senescent cells. But what we found is that if we look at markers of this mitochondrial apoptosis, so in this case, we use an antibody that detects an active form of BACs, BAC6A7, that is only present when these BACs and BAC macropores form. So if we look in a young proliferating cell, we could not see any staining in these cells. But then if we look at senescent cells, we found evidence for a group of mitochondria, very few mitochondria, that are usually in the periphery. They are circular, and they are positive for this BAC6A7. We then also did other methods to analyze this, including looking at the formation of BAC's oligomers. And we found that these were not present in proliferating cells, but only in senescent cells, consistent with the idea that these BAC's macropores were being formed in a senescent cell. Then we thought, okay, what happens if we delete BACs and BAC, stopping these cells to be able to form these pores? And what we found is that by doing so, we don't get the release of the mitochondrial DNA into the cytosol. And if we look at the SASP, we see that the SASP is suppressed, both at the mRNA level, but also at the protein level. So what I'm saying is that there is a subset of, a few mitochondria that are undergoing an apoptotic process in senescent cells. This is not sufficient to kill the cell, but is sufficient to release mitochondrial DNA, which will then drive the SASP. So we then did experiments in vivo. So we took aged mice and we deleted both BACs and BAC, and we showed that if we do that, we are able to reduce the SASP in these mice. We then tried to find ways that we could do this in a sort of pharmacological way. So we found drugs that are able to inhibit BACs, and particularly that BACs can actually go to the mitochondria, translocate to the mitochondria, and form these BACs and BAC pores. So we injected a drug called BAY1, which is a BACs inhibitor, into aged mice around 18 to 20 months of age. And what we found was quite striking. So these mice were doing better in all the health span parameters that we could find. So we looked at neuromuscular coordination using a method called rotarod. We look at their ability to stay in a pole for a long period of time. And we saw that by inhibiting apoptosis, we're actually able to see improvements in these mice. We looked at grip strength, found that these mice were doing better. We also looked at many parameters of frailty and found that the frailty index was improved in these mice. We also did CT scans of their bones and found that the bone microarchitecture was improved in these mice. and we saw that there was a decrease in inflammation in the bone of these mice as well. We also found that this drug penetrated the blood-brain barrier and actually reduced inflammation in the brain. And if we did single-cell RNA-seq, we saw that there was a reduction in SASP in both oligodendrocytes and microglia. So these are my conclusions from this data. So we think that our discovery shows that we have to stop looking at apoptosis and senescence as two separate cell fates. These are mechanisms that are actually regulated by similar mitochondrial dependent mechanisms. We think that this apoptosis occurring in a very few number of mitochondria that doesn't cause cell death is a driver of the SASP via the release of mitochondrial DNA into the cytosol. And surprisingly, even though the entire field of senescence is trying to kill senescent cells, I'm actually proposing that inhibition of apoptosis may be a way in which we can counteract aging and age-related disease. I just had a final kind of thought. This is something I just had yesterday, and I was putting together the presentation. Is you know, if you think about apoptosis, apoptosis is usually seen as a good thing. You know, you have a damaged cell, and then you have this process that is a kind of all or nothing. So usually what happens is that when a cell becomes apoptotic, all the mitochondria undergo this release of cytochrome C, mitochondrial DNA, and then you have cell death. So actually senescence is a failure of a cell to self-destruct, because there's only a few mitochondria that undergo this process. There is no cell death, but this failure leads to the SASP. Perhaps is a safe fail mechanism by which cells that are failing to become apoptotic may actually induce the SASP to recruit the immune system to eliminate them. And perhaps, you know, this failure to apoptose is what results in the accumulation of senescent cells as we age and the increase in inflammatory processes. Okay, and with these thoughts, I would like to finish, and I would like to acknowledge my lab was worked a lot for this story, particularly Stella Victorelli, but also other lab members that have really worked a lot for this project. And I would like to thank funding sources, the NIH, the Glenn Foundation, and I would like to thank you for inviting me and for your attention.
Thank you so much. What a wonderful talk, and congratulations to your very, very nice paper being published in Nature. So I think your group and yourself, you're very proud. So, and wonderful for data, really shaking up, I would say, the senescence field, because indeed senolytics and xenomorphics have are just a hype at the moment, and we are introducing them into clinical trials.
I know.
Yeah. Okay, but maybe we can touch on that a little bit in our discussion. First of all, I would like to ask you, I always learned that senescence is not senescence. Like senescence can be repetitive, can be stress-induced, etc. Have you looked at different inducers of senescence, and does that matter in your entire hypothesis, how mitochondria and senescent cells are interacting?
Yeah, I mean, so usually in my lab, I mean, obviously there are limitations. I mean, we use models that are established, you know, since the time of AFLIC. We mostly, you know, to use kind of fibroblasts, human fibroblasts as a model to study senescence, but we try to reproduce majority of the data that we have using different inducers, you know, DNA damaging agents or replicative senescence or oncogene-induced senescence. And particularly when it comes to mitochondria, in most cases, there are differences in function. They may not be all exactly the same. But this may be also a question of kinetics, because senescence is a phenotype that takes quite a long time to develop. So sometimes changes in mitochondria may occur early on, some other changes may occur later on. So it's well, obviously complicated to know and to compare the different models of senescence. But I would say that in terms of different stressors, we find this phenomenon of leakage of mitochondrial DNA is common across most ways that we have induced senescence in fibroblasts. So if we fractionate the cytosol of senescent cells induced by oncogenes, induced by stress, induced by replicative senescence, we basically see very similar phenotypes. We also see evidence for changes in mitochondrial function that are similar across senescent types. However, there are differences. Some of them, it may be that in some types of senescence, mitochondrial dynamics works in a way that is different from oncogene-induced senescence. But we still don't understand exactly why.
Yeah, understand. So I think Heiflich will like that I think we're now getting grip on why replicative senescence is maybe bad. But is it tissue dependent? Have you tried other cells than fibroblasts?
Obviously we have studied other cell types. Obviously, endothelial cells, we've looked at pre-adipocytes in collaboration with others, and many of the features of senescence are, particularly when it comes to mitochondrial dysfunction, are sort of common across cell types who we analyze. Difficulties that we have is really the in vivo environment. So, if we still haven't really found ways in which we can isolate senescent cells from, human tissues or mouse tissues and analyze them for mitochondrial function or other features of senescence. That's the field, obviously everybody's very excited about senescent cells. We've been studying senescence since the 1960s, but majority of the data that we have from senescence comes from in vitro models. And only now we seem to have the technology to be able to look in more detail at what happens to senescence in vivo. So I think that, your question is fair, and I think that the future will tell us a lot more about that.
Yeah, and especially if you look at different organ systems, different cell types within the organs, mitochondria might also play a different role based on the metabolism in these kind of cells. Can you hypothesize, or maybe you already tested it, how much contribution then the leakage of mitochondrial DNA, etc., has, and in the contribution to certain senescence phenotypes in different organ systems based on their metabolic play they have to play to get us as humans up and running?
That's a great question. Obviously, if you have certain tissues that have a lot of metabolic demands, like the muscle or the liver, they have a lot more mitochondria than others. Obviously, they accumulate senescent cells as well. But so far, we have really, from a technical perspective, has been really difficult to determine this mitochondrial DNA leakage at the cellular level in tissues. So we can detect it very easily in the blood. And there is, good data to indicate that you can find increased circulating mitochondrial DNA. In fact, it has been associated with aging and age-related diseases. And actually, there's evidence as well that if you treat mice with senolytic drugs, you can reduce the circulating mitochondrial DNA. However, the origin of this mitochondrial DNA, it's still not known. So this is something we're working on, but we still don't really know if there are differences in different tissues regarding leakage of mitochondrial DNA.
Let's stay a little bit at the leakage with the mitochondrial DNA. It's so fascinating. So you have studied, but maybe I missed it, that if there is mtDNA leakage within senescent cells, that then you have lots of SASP. Does it also occur in a state where cells are not senescent? And could that actually be a problem we see during the aging process of inflammation, etc.?
Yeah, I mean, obviously it is possible. I mean, we studied specifically in the context of senescence, and but it is possible that other cell types that are not. So what you're sort of suggesting is that during aging, you may have increased leakage of mitochondrial DNA, and this may be in fact preceding the presence of senescent cells by causing inflammation. So it is possible. I'm obviously determining chicken and egg is always complicated in these scenarios. So is it mitochondrial DNA coming from senescent cells, or is it mitochondrial DNA first causing inflammation, driving senescence? So that's a complicated question to answer. The only thing I could say is that there's at least one study that was led by a group in Harvard by Stefan Tullius that basically looked in aged mice treated with senolytic drugs. And by treating these mice with senolytic drugs, they saw a reduction in circulating mitochondrial DNA, which would suggest that the senescent cells may be major sources of this mitochondrial DNA. But obviously, the treatment that was made was with the dasatinib and quercetin. It's possible that they may have other functions or even protect mitochondria and do things that are not necessarily related with senescence. So there's always caveats in interpreting this data.
We always thought that senolytics are the way to go to treat aging processes. What I learned in your talk, maybe we should talk about mtDNA lytics. But now you are saying let's inhibit apoptosis. Can you share a little bit your ideas how that in maybe 10 or 20 years would possibly interfere with clinical treatments, etc.? And hearing your talk where you see a way to go forward with senolytics versus all the other options actually published.
I mean, this is a complicated question.
I know.
But obviously, there's many camps in the senescence field. I would say that the senolytic camp is basically saying, okay, this is probably one of the best ways to target senescence because it's the hit and run idea. So you don't need to take a drug all the time. You'd be able to take a drug, eliminate the senescent cells, and then you don't need to be taking these sort of SASP suppressors all the time. So from a clinical perspective, that's better. I would say that there are still things we don't know about senolytic drugs. One aspect that I think is really complicated is the fact that we see senescence or senescent markers accumulating in post-mitotic cells. So the idea, for instance, that if you have neurons that are expressing senescent markers and you treat them with senolytics and you are clearing them, is that necessarily a good thing? Is it good to lose some neurons or lose some cardiomyocytes or lose some, muscle fibers as a consequence of this? While mice may be able to regenerate, can humans regenerate those lost cells, particularly as we age? So that's an argument against senolytics. Xenomorphic argument, obviously, we need a SASP as well. I mean, obviously, the SASP plays important functions. So keeping the SASP and suppressing inflammation is really a good thing. And obviously, the argument here, is if you're stopping apoptosis, you're also stopping a mechanism that is essential to suppress cancer. So, but strangely enough, I should say that when we looked in our mice that we inhibited PAX and BAC, we actually did not see an increase in cancer incidence. So I think that, we need to investigate all possible mechanisms and do a lot of, careful preclinical studies. And if these drugs show any kind of advantage, then we can move into the clinic. As I say, this study is a proof of concept. We're still not in a stage where we can go into thinking of translational potential.
Absolutely. Thank you so much for your careful careful answer. We are going over to Jessica Liu, who is our PhD student at the Center for Health Longevity, and I see there are lots of questions from the audience. So over to you, Jessica.
Thank you, Professor Martin. Thank you, Professor Passos. So there's a related question, I think, with the last one is, and I'm going to combine two, but why might there be rogue mitochondria? How do they become these type of mitochondria? So what defines this subtype? And how come the other mitochondria are not affected by the activated BACs or BAC?
Okay, that's a great question. So we think, actually, I didn't really show this data, but one thing that we found is that in senescent cells, mitochondria start making very tight networks. So mitochondria can be, they're very dynamic organelles and they're all communicating with each other. But what we found is that in senescent cells, they actually form very tight networks. And these mitochondria that seem to be the dysfunctional ones are actually mitochondria that are separated from these networks. So we think that actually what happens is that as cells become senescent, there's possibly an adaptation for where a lot of the other mitochondria kind of group together, and so that they undergo fusion as a way for not all the mitochondria to start releasing the mitochondrial DNA and the cytochrome C in the cell. So as a mechanism that stops the cell from dying. So that, but obviously, there's a few mitochondria that potentially, because the cell is not able to remove them via mitophagy, they might simply accumulate and leak this mitochondrial DNA. So, and the reason we think that this fission may be important is that if we then take a senescent cell and then we inhibit fusion, so that in the end you get a lot of circular and dissociated fragmented mitochondria, we actually see a lot more leakage in the senescent cells and a much higher SAS.
Wow, that's really interesting. There's another question a viewer asked, and they asked, have you done a longer time for the BACS inhibitor treatment in mice? And in that case, have you observed any malignant transformation in them?
That's a great question. So we've done a lifespan with these mice. So we didn't see any evidence for increased cancer in this cohort, surprisingly. I was actually expecting that this may happen, but we did not see a significant increase in lifespan. So these mice, so we actually treated the mice until the end of their lives. And we saw that there were improvements in health span parameters as we follow these mice, but no increase in lifespan and no changes in cancer burden.
Wow. And would the goal be to eliminate all senescent cells, or would there be any side effects if all of them were eliminated at an older age? Because I know you mentioned in the beginning that it could be helpful at like younger ages.
Yeah, I mean, obviously senescent cells, I mean, I would say that, the senolytic approach, we're just eliminating senescent cells that are there, but we're not preventing cells from undergoing senescence. So if you have damaged cells, they still can become senescent, and if they are eliminated by the immune system, that's great. The senolytic approach would be, we're trying to tackle the senescent cells that are there, that are accumulating, and by eliminating them, I think that actually this may be beneficial in the context of cancer suppression, because there are studies that show that if you eliminate senescent cells, you actually stop development of cancer, because senescent cells can actually, if they persist, they can be pro-tumorigenic.
And then, so then in another group of individuals who may have mitochondrial myopathies, would they display greater accumulation of senescent cells or greater expression of SAS factors in circulation?
That's a great question. I think I've seen I have not seen many studies where people have looked into that. I think I'm not aware if this has really been studied. I've seen in a few conferences, I think I've seen in a mitochondrial conference that some people were studying that, but I'm not aware of anything that has been published lately. I know that, for instance, mouse models of mitochondrial mutations, like the Pol gamma mutator mouse. So this is a mouse model that has a polymerase, a defect in a polymerase and accumulates high burden of mitochondrial DNA mutations. And we looked in tissues from these mice, I think Judy Campisi as well, and saw an increased burden of senescent cells. So there's obviously some connection between mutation burden of mitochondrial DNA and senescence. But in the context of patients, I'm not entirely sure if there is very strong evidence at this stage, as far as I'm aware. But I may be wrong.
Wow. Now I'm going to ask a few questions that are kind of zoomed out about more kind of lifestyles and what can be applied to us. So there are questions about how, are there any lifestyle changes that have been shown to reduce the effect of senescence, or are there any supplements that would boost mitochondria action?
Right. Okay. So there are studies that have shown, particularly in mice, that I think as well in humans, that versus exercise may be a senolytic, not a senolytic, but it will reduce burden of senescent cells. So that is a lifestyle intervention that does have an impact in senescence. Obviously, caloric restriction has been shown to also reduce burden of senescent cells, at least in mice. There's obviously drugs that target different aspects of mitochondrial function that may have impacts on senescence. I mean, we've shown that in vitro, things like MitoQ and others can have impacts on the SASP, and also you can extend replicative lifespan of fibroblasts if you treat cells with MitoQ or other mitochondrial targeted antioxidants. But I think there is absolutely, as far as I'm aware, not really good evidence that any of them can be, at least these mitochondrial targeted drugs would bring any benefits so far in humans. And I would be very cautious about trying them.
So there was another viewer that was wondering, what would happen if a person doesn't develop any senescent cells as they age, or is that even possible to not develop any senescent cells?
That's a great question. I don't know if it is possible or not that somebody would not develop any senescence. I would think it's unlikely. But I mean, most of our studies characterizing senescence, I mean, so there's a lot of issues with characterization of senescence, I should say, is that we lack markers to identify senescent cells. That's one of the issues. So we've studied senescence in vitro for 40 years. Not me personally, but the community has studied senescence for many years. But we've studied senescence in vitro, and only now we're starting to look at senescence in vivo, and we are realizing that it's much more complicated than we thought initially. So I just actually wanted to give you an example. So I'm part of a network called CENET. And this is a common fund by the NIH. So the NIH is one of the big funding bodies here in the United States, and they are sponsoring a project involving many labs across the U.S. with the idea of making a map of senescent cells across different tissues during aging. And this is a project that is, similar in scope, like the Human Genome Project, and they have, they're spending, 250 or 300 million on it so far. So it's a big thing, but it feels like only now we actually have the technology, because with all these single cell technologies that we can look an individual cells and look at many different markers simultaneously, or you can look at transcriptome, proteome, epigenome of single cells. So now we can characterize a senescence in vivo. And I think this will tell us, what is happening as we age, what tissues accumulate senescent cells, and if, maybe there are some individuals that may have less propensity for senescence than others. And perhaps those are the longest lived. We don't know.
Amazing. And if the viewers want to know more about your research and what you're doing next, where can they find you the easiest? Or how?
You can email me. So, passos.joao@mayo.edu. So obviously my lab is interested in, so we are involved in many projects to do with mitochondria and the relation to senescence and aging. We also have telomere projects, which I haven't talked about today. So my lab does a lot of investigation into how telomeres contribute to aging. And we are very much involved in this CENET consortium where we are mapping senescent cells and developing new technology to map senescent cells. So, so if you're interested in learning more, please contact me.
Amazing.
Good. Perfect. I see that your email address is already in the chat, so you might regret it because there are lots of questions we have not answered and we couldn't answer because of the time limit we have. Thank you so much for this wonderful presentation, these really provoking statements for today. I think it really changed my view on senescence. Jessica, thank you so much for moderating. Thank you to be here tonight. You might be interested in other educational events we have, and that's in February 2024, not so far away from now. We have the Academy for Healthy Longevity, and the Academy is organizing the intensive course taking place at the 26th to the 29th of February 2024. And if you want to be in direct contact with international key opinion leaders, if you want to have a crash course about health and longevity in just 3 days in an inspiring environment, this course might be for you. And the intensive course is followed by our conference, Unlock Healthy Longevity with Supplements, taking place from the 29th of February and on the 1st of March in 2024. And we dedicate the conference entirely on supplements, but really including the preclinical research, the clinical research, the translation into clinical practice, hearing from practitioners, hearing from regulators to really discuss regulatory aspects. And there's also a startup competition this time. So make use of the early bird, I would say, and the discount is just happening until the end of this month. Next week, on the 19th of October, 2023, of course, this year, Professor Brian Kennedy is hosting a special guest, and that's Dan Budner. And he is a National Geographic Fellow, a New York Times best-selling author of 5 books on longevity and happiness. Happiness is very important, especially in Singapore. And we will talk about the Netflix documentary. And I think you heard it, that maybe Singapore is also a blue zone in how to live to 100. We are closing today not with the Netflix series, but we are closing with a video about also a very important topic, happiness, how to achieve a happy marriage. Take care.
You 103 years old?
Yeah.
And you 100.
Yes, sir.
You got to be the prettiest 100-year-old. They've been married 82 years. So now, Miss Willie, you said that the secret to a happy marriage is being nice to each other.
Yeah.
Any other advice you have?
Just sit down and talk things over.
Talk things over.
Oh, yeah, and get a good understanding of what you're doing and where you're going.
Let me ask you something. It's a lady up here that's been engaged. She been with her boyfriend 11 years.
Yeah.
They've been engaged for 5 years.
Yeah.
He want to get married. She don't want to get married because she think they ain't got enough money to have the big wedding she want.
Big wedding.
Yeah, big wedding, Mr. DW.
Well, now, why you want to bring us into that?
You don't need to have no big wedding. Just go and get married.
Don't need to have it, just go get married. Let me ask y'all a question.
Okay.
How long did you court her before you asked her to marry you?
Oh, about 3 months or so.
3 months?
Yeah.
When you're young, you don't know any better.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.